Today, we will elucidate a review article from Nature Reviews entitled Biomarkers of Aging of Humans and Non-Human Primates. This review summarizes recent advances in the study of aging biomarkers at the cellular, tissue, and organismal levels in both humans and non-human primates through a systematic analysis of representative biomarkers across diverse biological contexts and scales. It provides an in-depth discussion on the variability and conservation of physiological changes associated with aging. Furthermore, it clarifies the significant value of these biomarkers in the assessment of aging and the practice of interventional strategies. A pivotal reason for selecting non-human primates as experimental subjects lies in their high degree of similarity to humans in genetic background, physiological structure, and functional regulation. Such commonality renders non-human primates an ideal model for investigating human aging processes, thereby offering critical support for bridging the translational gap between fundamental research on aging mechanisms and clinical applications. Moreover, the lifespan of non-human primates is closer to that of humans, enabling the observation of long-term dynamic effects of aging on a timescale more analogous to human conditions. This advantage further underscores their irreplaceability in aging research. This article primarily elucidates biomarkers of aging at the cellular, tissue, and organismal levels, while also addressing certain multi-level biomarkers shared across these biological scales. The authors contend that establishing a unified research framework for primate aging biomarkers will not only provide more systematic theoretical support for deciphering the essence of aging, but will also lay a critical foundation for developing effective intervention strategies against age-related diseases and advancing the global goal of healthy aging. Subsequently, the discussion elaborates on representative biomarkers of aging at the cellular level. The figure on the left presents an overview of key cellular aging biomarkers, which span various subcellular and molecular processes such as cell cycle arrest, loss of genome integrity, telomere shortening, among others. The figure on the right further delineates specific representative cellular aging biomarkers corresponding to each of these subcellular and molecular mechanisms. Together, these illustrations provide a comprehensive elucidation of the characteristic cellular alterations and molecular regulatory networks underlying aging, while also highlighting their potential relevance for developing targeted therapeutic strategies against cellular senescence. We now turn to the exposition of biomarkers at the tissue level. In this section, the authors provide an in-depth exploration of the complex landscape of aging biomarkers across various tissues and organs in primates, highlighting both shared features and heterogeneity in the aging processes among different tissues. Through a detailed discussion of the skin, central nervous system, cardiovascular system, lungs, liver, immune system, adipose tissue, musculoskeletal system, kidneys, and reproductive system, a range of representative biomarkers are examined. These biomarkers reflect histological, structural, physiological, functional, cellular, and molecular alterations associated with aging. Biomarkers of aging at the organismal level provide a holistic perspective on the biological changes occurring during aging at the individual or population level. These biomarkers encompass a broad spectrum of alterations, including physiological changes in functional performance and morphological characteristics, intracellular molecular deficits, and dynamic shifts in the microbiome. This section examines two major categories of organismal aging biomarkers: conventional biomarkers and aging clocks. We now focus on aging clocks. Among various models for aging assessment, epigenetic clocks, particularly those based on DNA methylation, have emerged as a prominent research focus due to their reliance on methylation patterns at specific CPG sites. These clocks have been extensively applied to predict chronological age in humans and various non-human primates across cells, tissues, and whole organisms, offering mechanistic insights into aging from an epigenetic perspective. DNA methylation clocks are capable of not only estimating chronological age and lifespan, but also show promise in assessing disease risk and evaluating the efficacy of interventions. Beyond DNA methylation-based clocks, other molecular aging clocks include ATAC clocks, where ATAC denotes assay or transposes accessible chromatin, microRNA age predictors, transcriptomic clocks, proteomic clocks, metabolomic clocks, immune or inflammatory clocks, and microbiome clocks, among others. Leveraging deep learning algorithms, various phenotypic clocks derived from physiological or functional parameters have also emerged as indicative biomarkers of aging. For instance, models constructed from thermal facial imaging or 3D facial morphology may offer reliable, non-invasive biomarkers for predicting chronological age and chronic disease risk. Similarly, models assessing psychosocial changes based on social and behavioral data can be used to estimate both chronological age and perceived rate of aging. Other aging clocks based on imaging, clinical parameters, composite measures, or wearable sensor data have also been developed. Collectively, these diverse clocks hold significant promise for quantifying biological aging and evaluating the effectiveness of anti-aging interventions. Analysis of aging biomarkers in primates at the cellular, tissue, and organismal levels has revealed both context-specific or layer-restricted alterations, as well as conserved, multi-level changes. The left figure illustrates several biomarkers of aging that are shared across different biological tiers. For instance, the activation of endogenous retroviruses ERVs serves as a biomarker of aging at the cellular, tissue, and organismal levels. This finding contributes to a systematic understanding of how molecular changes progressively drive the aging process. ERVs can generate retrovirus-like particles that promote senescence in neighboring cells via paracrine signaling, thereby acting as senescence activators and accelerating local tissue aging. These particles can subsequently enter systemic circulation and propagate aging effects across multiple tissues, ultimately contributing to organismal aging. Furthermore, other senescence-associated factors are capable of amplifying aging signals in a similar manner, providing additional insights into how aging programs may be hierarchically initiated from the cellular level to tissues and ultimately to the whole organism. We now present the concluding in perspective section. First, an ideal biomarker of aging should meet the following criteria: high specificity and precision, demonstrating high specificity for aging processes, exhibiting a strong correlation with biological aging, and accurately predicting declines in health status. Stability and generality, showing good reproducibility and generalizability across diverse physiological or experimental conditions. Responsiveness and sensitivity, capable of reflecting both accelerated and decelerated aging, and responsive to interventions. Convenience and scalability, allowing minimally invasive and cost-effective measurement. Currently, such reliable and universally applicable biomarkers of aging are still lacking. Furthermore, the inherent heterogeneity of aging stemming from variations across cells, tissues, individuals, sexes, and even ethnic groups means that a single biomarker may be insufficient to accurately capture the complexity of aging. The nonlinear and multifaceted nature of aging, spanning multiple levels of biological hierarchy, further complicates the establishment of robust aging biomarkers. With the rapid advancement of high-throughput omics technologies and functional genetic screening, a vast amount of aging-related biological data has been generated. This includes molecular-level data with single-cell and spatial resolution, as well as large-scale datasets from cohort studies. In this context, improvements in artificial intelligence, AI algorithms, and models can help systematically integrate and interpret these complex data. AI is expected not only to deepen our understanding of the fundamental mechanisms of aging, but also to facilitate the identification and development of potential universal biomarkers for aging assessment and interventions targeting age-related diseases. The integration of multiple biomarkers and the development of multimodal aging clocks may offer promising avenues for addressing the complexity of aging prediction. However, the selection of optimal multifactorial combinations remains challenging. Moreover, comprehensive validation processes involving multifaceted assessment are essential to ensure the reliability, accuracy, and clinical utility of aging biomarkers. Given the genetic and physiological similarities between humans and non-human primates, NHPs, biomarker-dependent studies of aging and NHPs hold great promise for understanding, evaluating, and intervening in human aging and age-related diseases. Nevertheless, it is crucial to address how to enhance the translational efficiency of preclinical findings from NHPs to clinical settings, while ensuring safety and ethical compliance. That's all. Thanks for your listening.